So now it's my privilege to introduce Dr. Christina Ruby, who's on the faculty in psychology at the Indiana University of Pennsylvania, and her interest and expertise is in alcohol and animal models of alcoholism and addiction, and she's done a lot of work with long-term effects of adolescent alcohol exposure on, especially on circadian rhythms and sleep and disruptions of mammalian circadian timing by alcohol and stimulants. And today she's going to be speaking with us about time out, alcohol's effects on circadian rhythms and sleep. Christina?
Thanks so much. That was an excellent introduction. I'm actually a professor of biology. I didn't point that out, but psychology is just as appropriate, I would say. I have a background in both biology and psychology. So let me get this geared up here. Can everyone see this? Everyone's able to see my presentation? All right, excellent. So yes, as you mentioned, I am going to talk about alcohol's effects on circadian rhythms and sleep, which I've actually been studying since I was but a student, a PhD student back at Kent State University in Ohio. But what I'm going to focus on is some of the work that I've collaborated on with Scott that's actually on adolescent circadian rhythms. And I think, as I understood, I kind of took a look at people's profiles in this group. And as I understand, a lot of you look at kind of some adolescent and addiction related behaviors. So we're going to focus on adolescents. I could probably skip this slide, but I always like to include a little background on why we should care about alcoholism and addiction. I think everybody has a pretty good handle on why we should care. It's really, really important. This is worldwide data. If you look actually within sort of the United States and Canada, and if you kind of combine all of the neurological and psychiatric disorders together, they far sort of outnumber other disorders such as cancer, heart disease, etc. So really important to study this, especially now. It's hard to kind of go through a week, I think, without seeing some sort of news article related to people's heavy drinking patterns during the pandemic and how many people are dying from overdoses of opioids like fentanyl during the pandemic. So this is really sort of pandemic part 2. And it's been going on for quite some time. It's just being exacerbated. And that's all I'm going to say related to the pandemic. Now, you may or may not be familiar with circadian rhythms, but I'm going to give us a little bit of background on circadian rhythms. Like I like to tell my students, whenever we want to know what a gene does, what do we do with a gene? We disrupt it, right? We knock it out, or we knock it down, or we overexpress it. That same sort of, I guess, idea can be applied to circadian rhythms. And when you disrupt circadian rhythms, it's really, really bad for your health. Circadian rhythms can be disrupted sort of genetically. You may have advanced or delayed sleep phase syndrome. Those things tend to be genetic. You may have other genetic sleep and circadian disorders, or disruption of circadian rhythms can arise from the environment. Things like jet lag and shift work, repeated jet lag especially, and shift work are really detrimental to health in a lot of different ways. And you can kind of see all different things. In terms, I'm obviously going to focus on neurological and psychiatric components of circadian desynchrony, but you know, it's kind of, I dare you to think of any kind of psychiatric disorder or neurological disorder that doesn't have some component related to circadian and sleep disruption. I can't really think of one. In fact, there are very few that don't. So it's really important to study circadian rhythms, particularly if you're interested in mental health. So what are they? Well, circadian rhythms are endogenous rhythms that are around 24 hours in physiology and behavior. And they include the most obvious one, which is the sleep-wake cycle, or the sleep activity cycle, you might call it. But they also include a lot of other rhythms that we may be less appreciative of. Hormone release, probably the most obvious ones that are regulated by the circadian clock would be cortisol and melatonin, but many, many other hormones and neuroendocrine factors are controlled on a circadian basis, or released on a circadian basis. And in fact, their peripheral responses to these things are also circadian in nature too. Gene expression, and as I'm going to go over in a moment here, of course, the cellular circadian timekeeping mechanism involves transcriptional and translational feedback loops. But apart from just the mechanism itself, many genes in many different cells are actually controlled by the cellular circadian clock, which every cell in the body has. Even red blood cells that don't have a nucleus have their own version of a circadian clock. So it's very ubiquitous in the body, and it's obviously very important. So circadian rhythms are maintained by the suprachiasmatic nucleus. You can see this tiny little pink dot that's here in the hypothalamus. I'll refer to it as the SCN because that's a lot easier to say. And it's referred to as a clock. And the reason that we call it a clock is because it exhibits the properties of a clock. It oscillates intrinsically and autonomously. If you pop it out of the brain, it'll actually keep ticking for as long as it lives, as the tissue lives. It's temperature compensated. So in other words, it doesn't run slower on a cold day and faster on a hot day. That's really important. Otherwise, it wouldn't be able to keep time. And it exhibits the ability to be reset. So much like a wristwatch needs to be reset every now and again, our circadian rhythms need to be reset too. And that's done directly through the circadian clock in the SCN. We also refer to the SCN as the master pacemaker. And the reason that we call it this tiny little region of the brain is pretty mighty. It's small, but it's mighty. And the reason for this is because it's actually synchronizing all of those cells throughout the body and all of those tissues and all of the different brain regions, which I'll talk about a little bit. Circadian rhythms actually require daily synchronizing input via these Zeitgebers. And the most important one is photic information, or the light-dark cycle. Pretty obvious why that would be an important one to time our daily cycles. Non-photic inputs include things like exercise, sleep deprivation, novel environments, and social interactions, or as I like to say, they include anything else that sort of can oscillate on a daily basis that is salient in our environment. And these, again, are really important on a daily basis to synchronize our circadian rhythms. So how can we assess circadian rhythms? The easiest way to look at the phase of the SCN is by looking at circadian locomotor activity. And the reason that it is the easiest way to do it is because it's non-invasive. And so we look at locomotor rhythms above sort of these other rhythms, unless you're particularly interested in one of these. So to get you used to kind of looking at what this is called an actogram. We typically double plot our actograms. In other words, they're 48 hours. We may even triple plot our actograms because it makes it easier to see the rhythm. And each one of these sort of lines is one day, day one, day two, day three, and so on and so forth. And from these circadian locomotor activity rhythms, we can actually look at a number of different measures, which I'm going to go over later today. We've got onsets and offsets, pretty obvious, you know, the start of the activity and then the end of the activity each day. Alpha is referred to as the active phase length, or the subjective night in terms of nocturnal rodents, which I'll talk about. I will typically refer to the rest phase as the rest phase or sleep phase or the daytime. Rho is another way that it is referred to. Tau is the period length. So for an animal that's entrained to a 24-hour cycle, it should be exactly 24. So that'd be looking from onset to the next day's onset. If you're free running, such as how the animal is doing in the second half of this actogram that's shaded, a free running period is actually going to be a little bit different from exactly 24, and that's referred to as tau, or endogenous period. And then finally, phase shift, which is going to be really key to what I talk about. The phase shift is the difference in onsets before and after an experimental manipulation. In this case, so here's the dots that denote the onsets before this experiment took place. This is the time of the experiment. Here's the onsets after the experiment took place, and then you draw regression lines and extend those lines, and you compare where those onsets are occurring after versus when they were occurring before. And in this case, you can see there's sort of a rightward shift of those onsets, and that's indicative of a delay. In other words, the animal is waking up later than it had before this experiment. If we saw a leftward shift of the activity rhythms, the animal would be waking up earlier, and that would be an advance. Okay, so that kind of brings me into how our sort of advances and delays actually regulated. And I mentioned that, you know, photic and non-photic inputs. So we'll start with the photic inputs because arguably, again, they're the most important. And we're looking at the black line on this graph here. The positive numbers denote an advance phase shift, which means, again, waking up earlier after the stimulation. And negative numbers denote a delay shift that's occurring due to the exposure to whatever input you're looking at. So during the nighttime, this is a version for a human being of these phase response curves. If you look at the black line, during the nighttime, if we're exposed to light early in the night, it's going to cause a delay shift. And the way that I like to sort of explain this would be like the sun going down later. So when the sun goes down later, it's a signal to our bodies to delay the next day. Apart from any, you know, effects on sleep, it does not depend on sleep. If, however, you're exposed to light late in the night or really early in the morning, you're going to have an advance shift. That's like the sun coming up earlier. And so it kind of makes sense that you would want to synchronize your rhythms to be a little bit earlier in relation to that. As you can see from the dotted line, this is non-photic phase response curve for a human being. This is exogenous melatonin administration. And it looks very different from the responses to photic input because at different times of day, it has its effects. So if you know anything about melatonin, it's really, it peaks in the middle of the night for all mammals. And it really, its point is to kind of measure how long the night is. And so if you administer melatonin in the middle of the day, you're going to have a nice phase advance shift because you're kind of tricking the clock into thinking it's earlier than it actually is. If you give melatonin later on in the night or sort of early in the morning, you're going to kind of trick the clock into thinking that it's later than it actually is, and you're going to cause a phase delay. And something that's important is to mention here is that all mammals show a similar pattern regardless of phase preference. So when I talk about phase preference, I'm talking about whether an animal is diurnal or nocturnal. We respond to light exactly the same, and we respond to exogenous melatonin and other non-photic cues exactly the same way. So in terms of neurochemical regulation of circadian timing, the SCN receives a number of different inputs. The eye, of course, when we shine light on the eye, we've got a monosynaptic projection from the retinohypothalamic tract directly from the retina into the SCN, and it communicates that light input via glutamate and PACAP. For non-photic stimulation, we use both serotonin and neuropeptide Y. Serotonin comes, of course, from the median raphe, and neuropeptide Y comes from the intergeniculate leaflet of the thalamus. And you can see, like most things the thalamus does, it's collecting information both from the eye and from the raphe to make sure that when it releases that MPY, it's doing it properly. So at the cellular level, as I mentioned, circadian timekeeping is accomplished by a series of transcriptional translational feedback loops. And they work exactly like the gears in a clock. So we'll start here with this heterodimer of BMAL1 and clock. BMAL1 and clock form this heterodimer, and they bind to E-box elements in the promoter regions of multiple clock genes. So on the positive end of the loop, you can kind of look at it as this little tiny gear here. We have ROR alpha and reverb alpha that actually control the transcription of BMAL itself. On the negative end of the loop, we have period and cryptochrome genes, which are transcribed and then translated. After they're translated, they actually get phosphorylated and marked for destruction, but at a certain point in the day, they accumulate to levels that are too high to phosphorylate and degrade, and they end up forming a negative repressive complex with their phosphorylator, casein kinase 1 epsilon delta. They locate back into the nucleus, and they repress their own transcription. A couple of notes that I want to make here. As I mentioned, each cell in the body has a cellular circadian timekeeping mechanism utilizing these genes or some, similar genes depending on the tissue. The other thing that I want to mention here is clock-controlled genes. So aside from just the clockwork itself, there are many different genes in certain cells and certain tissues that are controlled by BMEL and clock, which means that they're going to oscillate on a circadian basis. In addition to just these clock-controlled genes, this is one of my favorite phrases, clock is a hat. Clock is a histone acetyltransferase, so there's actually rhythms in acetylation and deacetylation that are occurring on a daily basis. And anywhere between sort of 3 to 30% of the gene products in a given cell can oscillate with the time of day. So pretty important. Okay, so the effects of light at the molecular level, I'm introducing this really to introduce why alcohol might mess things up in the SCN. So at the molecular level, I mentioned that glutamate is the neurotransmitter and PACAP is the neuropeptide that are responsible for photic phase resetting. Well, glutamate is released and PACAP is released, and they have this postsynaptic response of course, activating NMDA-mediated signaling, activating CREB, and lo and behold, PER1 and PER2, in addition to that E-box that they have on their promoter regions, also have a CRE. And so CREB is actually responsible for phase resetting by light in the SCN. Something else that I'd like to point out here is that the subunit of the NMDA receptor that is responsible for photic phase resetting is the NR2B subunit, which, as we know, is famously ethanol sensitive. So I guess you can kind of see where I'm going with this talk, is that how alcohol might interact with photic phase resetting and some other aspects of circadian rhythms. Okay, so let's get into alcohol a little bit. As you might imagine, alcohol is not good for our circadian rhythms. And circadian rhythms, as I'm going to talk about, can also influence drinking patterns and other things as well. But alcohol is very highly and profoundly disruptive to circadian rhythms. It delays and reduces daily activity. No big surprise there to anyone who's ever tried a beer or two. It shortens free running period. It alters the balance between slow wave and REM sleep, and it alters sleep latency. Depending on if it's on board or not on board, you're going to have reduced sleep latency. In other words, it's going to make you fall asleep faster if you're drinking. But if you're in withdrawal or if you are an abstinent alcoholic, you're going to have problems getting to sleep and maybe staying asleep. In addition, it blunts and delays hormonal and clock gene rhythms as well in a couple of areas of the brain and body. And it blinds the clock to light. That's kind of related to that previous slide that I was talking about, where ethanol interacts with glutamate signaling in such a way that it essentially blinds the clock. Circadian timing, as I mentioned, also can influence drinking, and this kind of goes back to that slide on why we should care about circadian rhythms. Environmental perturbations of circadian rhythms like shift work, jet lag, or even genetic sort of circadian sleep disorders can greatly increase the risk of alcoholism and addiction in a number, in addition to a number of other disorders. Insomnia during protracted abstinence can definitely promote relapse, and in really severe alcoholics, I mean, they have a hard time sleeping at all. And that can just drive them nuts and lead them to drink more. Clock genes are also associated with alcoholism in both humans and rodent models. And when I say clock genes, I don't mean just a handful. I mean pretty much all of the clock genes that I went over in that slide where I was talking about the clock gene machinery. almost all of them are related to alcoholism in humans, and many in rodent models as well. PER2, I think, is the one that people might be familiar with. It causes advanced late phase syndrome in humans, and then it also, in rodent models, if you knock out PER2, you get a very alcoholic looking mouse. And they have problems with glutamate uptake, of all things. So the only problem with all of this stuff that I just told you is that I might be lying. Most of the studies have only examined adult males. And so we really don't know very much about females, and we really don't know much about adolescents or even really aged animals. So it's really important to kind of look at these other ages and sex differences as well, which I'll talk about for the rest of my talk. I know I'm preaching to the choir here, probably. Why should we look at adolescents? Well, early exposure to alcohol is extremely common. And when I say alcohol, a lot of alcohol, up to 10 drinks and 15 drinks on a single occasion, that's a lot of drinking. Very, very heavy drinking in high school seniors especially, and it's probably not better now. We also know that early alcohol use has some pretty profound neurobehavioral consequences. It certainly increases the long-term risk for addiction to alcohol and potentially even other drugs. It impairs multiple sort of levels of cognition and decision-making, attention, verbal learning, spatial processing, and memory. And we know that it alters structural development in the brain in terms of gray and white matter. In terms of circadian rhythms, what we know is that human adolescents tend to be naturally night owls. And the kind of issue with that is not so much that their circadian rhythms are delayed, but they're delayed in relation to their school start times. And we kind of impose all sorts of disruptive influence over this natural progression to being a night owl, right? But in addition to this, what's interesting about just being a night owl, whether you're an adolescent or not, is this eveningness phenotype is very predictive of alcohol and drug use. And it's especially predictive in youth populations. Being a night owl is also associated with kind of what we might think of as an adolescent phenotype, behavioral phenotype, like novelty seeking, impulsivity, and reward dependence are often associated with, being awake and alert in the evening as opposed to the morning. We don't really know why. In fact, there's a lot that's just left unanswered there, but that's exactly why we should be studying it. Again, preaching to the choir here, we're probably pretty familiar with being able to model aspects of alcoholism and other disorders with rodents. But it's really been kind of great to see how translatable the studies using rodents are to the human adolescents as well. This is a lot of sort of Scott's work over decades that I'm talking about here. Adolescent rodents, we know, are differentially sensitive to alcohol. In some aspects, they're less sensitive, and in other aspects, they're more sensitive. So we know they have a reduced sensitivity to sedation, but they have greater sensitivity to some of the fun effects of alcohol, like social facilitation and maybe anxiolysis. But they also have a greater memory impairment at the same dose compared to adults. That's a really bad combination, as I like to always point out. Not knowing you're getting drunk, and then also having the fun aspects of alcohol and greater memory impairments is a really bad combination. They also show higher alcohol consumption and heavy binge drinking, kind of like what we saw with human adolescents. Scott's recent studies and my recent studies have to do more with adolescent intermittent ethanol use and AIE, but this AIE exposure tends to lock in this adolescent phenotype into adulthood. And that refers to those responses to alcohol, so the sensitivity is sort of locked in, doesn't look like an adult, it looks more like an adolescent, and the heavier drinking patterns as well. Cognitive flexibility is reduced, and you can see kind of behavioral inhibition is reduced as well. We have increased anxiety. We have some sleep issues, like an increase in sleep latency and a reduction in slow wave sleep, at least in rat models. And we've got impulsivity and risk-taking being pretty high as well. So a very adolescent-like phenotype, and in particular with relation to alcohol. In terms of the mechanisms, we see decreased neurogenesis, increased neuroinflammation, and epigenetic alterations. The Nadia has really done a lot to kind of understand the mechanisms underlying these behavioral consequences. The big question is, though, in relation to circadian rhythms, we don't know anything about circadian rhythms in adolescent rodent models. We hardly know anything about circadian rhythms and their regulation in adolescent humans, let alone the effects of alcohol. So figured this was a really good thing to start asking questions about. And this brings me to sort of the first paper that I collaborated on with Scott, where we were addressing this question of, do adolescents differ in their circadian responses to ethanol? So here was the experimental procedure. What we did was we had a group of adolescents, and these are all males, so I apologize that they're all males. I am going to look at sex differences in the next paper a little bit, but it was just easier to keep one sex, so we just happen to have males. So we've got our adolescent males. These are actually kind of weanlings. We let them grow up a little bit before we start looking at the data. We've got adolescents and adults that are acclimated to our testing room, our circadian room. We then look at their entrainment to LD, that refers to the light-dark cycle, and it's a typical 12-12 light-dark cycle. So we look at that for sort of the first 20 or so days. We then inject them with alcohol, and the dose we use of alcohol is actually fairly low for mice. It's 1.5 grams per kilogram, which is equivalent for a human to about 3 drinks, hence why I have the little beer pyramid there. So we either injected them with alcohol or saline, and then we gave them a light pulse immediately after that injection. We do our injections in dim red light, and then we do the light pulse immediately after that. We then release the animals into constant darkness, and you can kind of see the same notion here. This is just a different kind of typical way a circadian researcher would represent an experiment. We've got the animals in a light-dark cycle for 14 days, and we looked at their entrainment to that light-dark cycle on the day of the experiment. We gave an injection prior to a phase-delaying light pulse, and then we released the animals into constant darkness for a couple of weeks. So in terms of what we found that adolescents have pretty high amplitude rhythms, and they actually have an advanced phase. So they had no problem in training. Adolescents and adults entrained to the same degree, but their amplitude of the rhythm was higher in adolescents. They're more active than adults. That's not a big surprise. What we did find was kind of interesting is that the adolescents woke up earlier. If you look at this 0 line, what we're comparing it to is the 0 line is the timing of lights out, and a negative value indicates after lights out. So if we see our adolescents, they're waking up pretty much a few minutes after those lights go out. Our adults, on the other hand, it takes them about a half an hour to wake up after those lights go out. In terms of offsets, we also see an earlier offset in our adolescents compared to our adults. The 0 line is the time of lights on, and the adolescents are actually sort of ending their activity before the lights even go on, whereas the adults are about half an hour into the lights going on that they actually end their activity. Alpha was actually a little bit shorter in adolescents, so that's actually somewhat potentially related to humans in adolescence tend to need a little bit more sleep than those of us in adulthood. In terms of the circadian rhythms and the entrainment, maybe mice are not really modeling that delayed phase that we see in human adolescence. And in fact, rats seem to model it a little bit better. Now, that being said, this next thing that I'm going to show you, which is the responses to light, I have reason to believe is pretty translatable to human beings. So what we see with our adolescents compared to our adults, I mentioned that alcohol sort of blinds the clock to light, or it reduces photic phase resetting. And we saw that with our adult animals that were exposed to this one dose of alcohol. They had only about half an hour delay in response to that light pulse, whereas those given saline had about an hour, a little bit more than an hour of a delay. So a nice healthy delay in the saline and an inhibited delay in the alcohol treated animals. On the other hand, for our adolescents, you can see alcohol did nothing, and that their baseline shifts were actually greater than the baseline shifts in adults. Now, this is actually pretty translatable for a couple of reasons, one of which is that we think that adolescent humans may actually be more responsive to the phase delaying effects of light compared to the phase advancing effects of light. So that may be pretty translatable. The other thing is that they're pretty insensitive to ethanol, at least at that lower dose. And that's also pretty translatable to what we've seen in some cases with ethanol research in adolescents. And this is just representative actograms of everything that I just said. So we've got our adolescents on sort of the left pane here, and our adults on the right pane, saline treated animals here and ethanol treated animals here. And you can see we got healthy shifts out of saline treated and ethanol treated adolescents, a healthy shift out of our saline treated adult, and not much of a shift out of that ethanol treated adult. The last thing that I want to make a point of is if you kind of take a look again at this free running period. So this is the point at which the animals were released into constant darkness. That shows the endogenous circadian period. And you can kind of see, especially in this top adolescent versus these two adults, but even a little bit in this guy here, they tend to wake up earlier and earlier and earlier in constant conditions, whereas our adults were waking up later and later and later in constant conditions. And so I decided, well, you know, that's really consistent between animals and between all the groups that I've tested. So why don't we, you know, measure it? And so we did, and we simply found that adolescents have a shorter circadian period than do adults. And again, the amplitude was higher in adolescents compared to adults. It may have nothing really to do with alcohol-related behavior, but I just thought it was an interesting change that was sort of age-related, that really had nothing to do with alcohol. So moving on to our next sort of set of research questions, I mentioned that I was interested in sex differences as well. Obviously, I used all males in that previous study. But even more, I was interested in looking at an AIE model. We wanted to know, you know, does adolescent ethanol exposure lead to lasting changes in circadian rhythm regulation, even after sort of a long period of abstinence? And then, of course, the sex differences. So the way that we sort of tested this was, I just had a group of sort of weanlings through adolescence that I acclimated to the chamber. We then exposed them as males and females in this case. We didn't have an adult control group. I would have loved to have done that, but it would have taken twice as long, and we just simply didn't have the space. We can only test like 24 animals at once. But so we decided to do all adolescents of different sexes. We exposed them to AIE or control for 24 days. Alcohol was available every other day, and it was 15%. They also had it in free choice with water. And on the days that they didn't have alcohol, they just had the water. Control animals just had water, and that was it for that period of 24 days. We then followed that with a 24-day washout period. And then on the day of the experiment, the animals are about that 10 weeks of age, just like what we saw before with those males that I did in the acute experiment. We gave them a dose of ethanol, an ethanol challenge dose, same dose that we used before, even though it's a fairly low one. It's about 3 drinks. Or we gave them saline, and then we followed it by a light pulse, a phase delaying light pulse. The no light pulse is just a control condition to make sure that ethanol doesn't have a phase resetting effect on its own, which it does not. I'm not going to go over those results. It's just kind of what you have to do to make sure you get published, is make sure ethanol doesn't have an effect on its own. So after this, we of course release the animals into constant darkness for a couple of weeks, and then we look at their activity. So no surprises here. This is very consistent with all the other literature that's looking at sex differences in alcohol and a lot of other drugs as well. Our females are higher consumers than our males of ethanol. They're drinking quite a bit, about on average 22 grams per kilogram per day. And I do actually control for leakage and evaporation, so that's the real deal. Our males are drinking quite a bit as well. There wasn't a difference in preference, as you can see. There's maybe a trend toward higher preference in females, but both males and females definitely prefer alcohol, even 15% alcohol, to water, because they're over 50% there. And all of the rest of the graphs that I'm going to show you, other than when I point out the next time period, are going to be from this AIE and control time period. So we're going to look at circadian rhythms during that period and then after. So this is during the AIE exposure. We saw sex differences, but not really AIE related differences in circadian rhythms in adolescence. And basically it can be summed up to say that the females are more active than the males. That's also pretty consistent with other literature outside of the circadian field. The females are just, they're a little bit hyper. They have higher amplitude rhythms. They show a little bit of a longer alpha or active phase. They're waking up earlier and they're going to bed later. And we don't really see any profound effects of AIE on any of those aspects. Females also, this is something that I did that was a little bit different from what I showed you with the adolescent and adult males. But the females actually showed more consolidated and robust rhythms during the active phase. So I actually looked at nightly activity and daily activity separately as well, just because I wanted to see what is our active phase and what does our rest phase look like. So the bout number was less in females, but they had less bouts, but they had more active bouts. So it's indicative of a consolidated and more higher amplitude rhythm. There's nightly activity and then in minutes, and then also counts, and you can see the females are sort of blowing the males out of the water in terms of how active they are during the nighttime. And again, there's not really an effect of AIE at all. Females also showed more rest phase activity, which might be indicative that they're staying up a little bit more. They're more active during the time that they would normally be sleeping. And something that I would point out here is that we really didn't have any evidence for any kind of alcohol or AIE-induced sleep disruption during the entire period of AIE. So there's not really any profound differences in AIE versus control animals of either sex. However, what we wanted to do, in addition to just looking at the group and that time as a whole, is compare the days on alcohol to the days off alcohol when we were looking at circadian rhythms in just that AIE group. So if you look at this, you can see that the alpha, while the animals were on alcohol, was actually a little bit longer than it was when they were off alcohol. The onsets were about the same in males. And I know this doesn't look like it's significant, but these are pairwise comparisons. So it's a very slight, a couple of minutes difference in onset when the animals are off alcohol. They're sleeping in a little bit, I guess. In terms of offset, you can see there's a pretty profound effect actually compared from the days on alcohol to the days off alcohol, with the days on alcohol being related to a later offset, and when they're off alcohol, we've got an earlier offset. That's actually kind of a little bit different than what we might expect, right, in terms of how it would affect sleep latency. And it's different from the I guess, work that's been done on adults as well. No surprise here, alcohol reduced nightly activity when it was on board compared to when it was not available. And that was significant in terms of the duration and the time. in general, and then we maintained those same sex differences that we saw before. Interestingly, we had no effect on rest phase activity when we compared the days on alcohol to the days off alcohol. And literally, it almost looks like exactly the same data. But I assure you, these were days on alcohol versus off alcohol. So again, even though they were drinking pretty heavily, there was really no evidence for alcohol-induced sleep disruption that we saw while alcohol was on board versus when it wasn't. So that's pretty interesting. So at this point, we're looking at the washout phase, and I'm not, rather than sort of like going over each little aspect of circadian rhythms, because they're pretty much the same as what we saw in adolescence with the sex differences and not really any effect of AIE. We did find one effect, and a pretty notable one at that. This is during the washout phase. Again, the animals are adults and they haven't had alcohol for quite some time. Our females actually showed evidence of an increased sleep latency. So the offset, again, in relation to the lights on, as you can see in alcohol naive animals, was earlier than it was in the alcohol exposed or AIE exposed animals. And so they were actually sort of going to bed later. At least, we didn't actually measure brain waves to look at sleep per se, but we can certainly tell a lot from this latency to enter the rest phase here or stop being active. So I think that's pretty interesting. It's about a 15 or 20 minute delay, and some of them obviously delayed a lot more than others. Finally, what we found was kind of the main point, which we were looking at alcohol's typical effect of blocking or attenuating photic phase resetting. And what we thought was we would get sort of a lock-in effect with our males, and indeed we did. If you take a look at this top graph, this is where the males are. And you can see that saline-treated AIE or control males had about the same shift, about an hour and a half of a delay. But if you compare that to those that received the alcohol challenge, you can see that the alcohol naive animals showed the expected attenuation that I've seen before with adult males, but those that got AIE had that lasting insensitivity. So it's as if the alcohol just didn't really impact them at all. And interestingly enough, if you look at this bottom graph of females, usually a graph without any significance isn't too interesting, right? But I found it quite interesting that none of the females had any sort of response to that dose of alcohol, which again is about 3 drinks. That's actually pretty consistent with a lot of other studies suggesting that females are less sensitive in some respects to some of the intoxicating effects of alcohol compared to males. So we're thinking that maybe there's a rightward shift of the dose response in terms of female animals. Okay, so in summary, hopefully you were able to kind of follow that along pretty well. In summary, our baseline circadian rhythms in mice were differing based on age and sex. We saw that our adolescents and our females especially have more robust and advanced daily activity. That was pretty clear. That's just baseline changes in circadian rhythms. In terms of their responses to alcohol, alcohol affects adolescent circadian rhythms differently. As we see with many other sort of neurobehavioral consequences, alcohol is different with adolescents. Our males were actually insensitive to that blocking of photic phase resetting by acute alcohol. And we really didn't see a lot of evidence for sleep disruption. And this may be related to using mice instead of rats. It may be related to allowing them to drink as opposed to administering alcohol. It could be just that they are less sensitive to sleep, or that that's not sort of something that always happens with alcohol. And then we found that AIE had lasting effects on circadian rhythms that did differ pretty profoundly based on sex, actually. So our females seem to have this delayed onset of their rest phase or sleep phase, which we think is indicative of an increased sleep latency. And our males remained insensitive to the effects of acute alcohol on blocking photic phase resetting. So all of these pretty interesting. In terms of future directions where I would go from here, certainly I would like to look at mechanisms. I mean, everything that I've shown you is really behavioral in nature, but I'd like to know why did we see these changes that we saw. As I mentioned, even with human adolescents, we're thinking that maybe they respond differentially to photic input as well as to alcohol. And I think a really good candidate is glutamate signaling in the circadian clock in the SCN. And the reason that I say this is because, of course, glutamate signaling is what's communicating photic information, but there's precedent for thinking, okay, adolescents may have a little bit higher excitation to inhibition balance in the brain. And there's precedent for that in other brain regions as well. So it would not be shocking, I think, to see that they have sort of either more glutamate release or a greater postsynaptic response to glutamate compared to adults. I'd also like to, of course, look at circadian clock gene expression. And of course, outside of just the circadian clock gene expression in the SCN, looking at the expression of other genes in other brain regions that are more, I guess, directly related to addictive behavior, directly controlling addictive behavior. I've looked at circadian clock rhythms, circadian clock gene rhythms outside of the SCN before, actually during my postdoc. Found some interesting things with, I don't know if you guys have ever heard the ENT1 null mice. It's an equilibrative nucleoside transporter for adenosine. You probably have heard of adenosine regulating sleep, but that ENT1 is actually also regulated by circadian rhythms, and adenosine itself can regulate circadian rhythms. So that's some crosstalk between the circadian and homeostatic sleep systems. But those mice, which have kind of a low adenosine tone, also have sort of arrhythmic and blunted period 2 expression in the striatum and in other regions of the brain, like the prefrontal cortex. So looking at clock gene expression in the SCN and other regions as well. And even just looking at circadian rhythms in other types of genes that really aren't part of the clock machinery. Females and dose responses, obviously that acute dose of alcohol did nothing to our females. They had no idea that it was there, and they're clearly able to drink a whole lot more than the males. So it'll be interesting to do more behavioral studies. So I showed you, again, and a lot of the research on AIE, as Scott can tell you, has been done on AIE, followed by a sort of washout period. But what happens if you never stop drinking? You know, what are the consequences of continued drinking on behavior and on signaling? We don't really know that yet. We got to take it piece by piece, right? So if you combined AIE and CIE, which is the adult version, I guess, of AIE, it'd be really interesting to revisit these questions. Non-photic phase resetting. So there's lots of reasons to look at this outside of just circadian rhythms. We know it's important that, you know, these social interactions, novel environments, and sleep deprivation can shift circadian rhythms, but they're also very translationally relevant to adolescence, right? Social interactions are absolutely key to development, especially during adolescence. Novelty is a big deal during adolescence, and sleep deprivation is something that we put our adolescents through a lot. So those have lots of sort of overlap in terms of translatability. And last, kind of alluded to this before, but age and sex effects on circadian rhythms in alcohol sensitivity and reward. So looking at behaviors that might not necessarily be regulated directly by the SCN, but regulated by other circuits that are influenced by circadian rhythms. We actually know that there's lots of alcohol sensitivity rhythms that are out there. So there's, you know, circadian rhythms in ataxia. There's circadian rhythms in loss of writing reflex. There's certainly circadian rhythms in drinking. If you give an animal 24-hour access to alcohol, they're going to show a pretty good rhythm in alcohol consumption. reward, there's a little bit less known in terms of whether there's circadian rhythms in how rewarding alcohol and other drugs are, but it's definitely worth looking at. And I think actually it's been done in some other instances, not so much with alcohol. And that brings me sort of to my final slide here. And this is just to point out all the oscillatory activity that's occurring throughout the brain. This is obviously a rat's brain, a sagittal section of a rat's brain. And these different colors are signifying how autonomous the oscillators actually are. And we're talking about clock gene expression as well as activity. Obviously, the SCN, the olfactory bulb, and the retina that are in this sort of orangey, peachy color, these are autonomous oscillators. In other words, you pop them out of the brain and you can keep them alive and ticking in a slice preparation without any outside input. In blue, we have the habenula, the dorsomedial hypothalamus, and the arcuate. These are semi-autonomous. So if you pop these out of the brain and you keep them alive in a slice, they're going to continue ticking for a while, but eventually they're going to stop having circadian activity without any input. And then finally, all of these other regions that I'm sure a lot of you are actually studying that are important, more important, I think, directly related to addictive behavior, such as the VTA, the nucleus accumbens, the amygdala, hippocampus, and dentite gyrus, the cortex, and so on and so forth. These all have clock gene rhythms as well, robust clock gene rhythms. But if you pop them out in a slice preparation, their clock gene rhythms are going to disappear without that outside input. So the reason that I present this idea is because even with an absence of any timing issues with the SCN or even changes in circadian locomotor activity, you may have changes in sort of clock gene expression or the workings of these other regions that sort of uncouple the SCN from these target regions. And I think that's a really interesting idea. That's a big, big question. And if anyone's interested in helping me answer it, I would love that because I can't do it by myself. But certainly I think it's a really interesting, and we have to look at, more than just the circadian activity, but these rhythms that might be more important and more directly related to addiction and alcoholism. And with that, I just want to say thank you so much for sitting through that. I want to thank Becky for inviting me. This is great. I want to thank Scott. It's so nice to see you. It's been a really long time. And thank you for your continued mentorship and just helping me understand this adolescent alcohol world, because it's a whole different world. And I want to thank, obviously, my funders here. And my students, especially Janella here. I love this picture of us, and this is what we do. We do all of our studies in the dark, right? This is circadian rhythms. Janella, who's here, she actually led that later study on the sex differences. She did an absolutely excellent job. Caitlin, who's shown here, she actually led the acute alcohol study, and then all these other students kind of contributed. It was pretty labor intensive, so I had a lot of students that helped out with these projects. Katie and Aubrey were actually my grad students that just recently graduated, and they were looking at combining drugs of abuse together. So we looked at alcohol, caffeine, and nicotine together. So it was a little bit of a separate project, but they're still sort of involved in this overall theme. So thanks so much. I hope we have time for a couple of questions. I know we're kind of running late.
We have a few minutes left, and I apologize for the technical glitches, but that was a...
Oh, it's not your fault. That's just the nature of meetings these days, right?
So I see in the chat that Amir Razvani has a question. Amir, do you want to unmute and ask the question?
Yeah, thank you for the talk. My question is there any information in circadian rhythm in alcohol-preferring rats?
Yes, actually, the researcher that studied primarily rats is Alan Rosenwasser. Do you know him? From the University of Maine in Orono, Maine. He's done a lot of studies looking at circadian rhythms in alcohol-preferring rats. He's done rats. I think primarily he's done rats, and he's done different alcohol preferring and non-preferring models. And there are circadian rhythm differences between those as well. He's also done some similar studies looking at photic phase responses and non-photic and things like that. So yes, that information is out there. And yes, they do look different in alcohol preferring animals than in non-preferring.
Have other questions? Well, I had a question. You did some great job in terms of the effects of adolescent alcohol on circadian rhythms, but what about having alcohol intoxication at different times of the diurnal cycle? Does that make a big difference?
It potentially could, and that's kind of going into why I think it's important to look at circadian rhythms in even intoxication and in sensitivity rhythms as well. I guess what I'd say is you could potentially model that in a lot of different ways, like give morning alcohol versus evening alcohol, or relative. I guess it would be more like early night versus late night for a nocturnal animal. And so I've had some students that are also interested in doing that too. You know, what happens if they're drinking kind of like human beings do, where, you know, late at night and kind of disrupting their sleep, versus if they drink when they would normally choose to drink, which is they wake up and they start drinking. And in fact, their highest alcohol consumption is something like 2 to 4 to 5 hours into the dark phase. So they pretty much wake up and hit the bottle right away, which pretty interesting. I mean, certainly I think, you know, if we didn't have work schedules and we were prone to addiction, maybe people would do it too. But yeah, that's actually a really great idea for a set of experiments as well. And certainly something that my students have suggested, because they said, well, when we drink, we do it late at night and we do it until 2 in the morning. So what happens then versus, you know, I guess Miller time at 5 p.m.
I see.
Yeah. So certainly I would expect different effects.
Okay. Anyone else have a point or a question? Well, I see it's straight up at a little past 1. So thanks again so much for your great talk. And it's the time of day is critically important for all of us. So thanks for taking time out of your day. And
oh, thank you so much for having me. It was great, and I can't wait to actually show up in person one time.
Yeah, we'll get through this soon enough. Okay, thanks a lot.
Right.
Thank you so much.
Great job, Christina. Thank you.
Thanks, Scott.